Chain termination or dideoxy procedure, it is the next method of DNA sequencing, and this method replaced the chemical method of Maxim and Gilbert because of its utility, and it can be performed for large-scale DNA sequencing. And this method was developed by Sanger and co-workers in 1977. And this method, it is used in conjunction with M13 MP vectors. अगर आपको याद हो कि M13 MP vectors जो हैं, they are basically single stranded vectors, और इनको यूज़ किया जा सकता है single strand formation के लिए, सिंगल स्ट्रैंडेड डीएनए की एम्प्लीफिकेशन के लिए, जिनको बाद में डीएनए सीक्वेंसिंग के लिए यूज़ किया जा सकता है. तो हम ये कह सकते हैं कि M13 MP जो वेक्टर की सीरीज़ है, ये एक सोर्स है सिंगल स्ट्रैंडेड डीएनए को हासिल करने का, और ये जो सिंगल स्ट्रैंडेड डीएनए है, इनको बाद वाले स्टेप में डीएनए सीक्वेंसिंग के लिए इस्तेमाल किया जा सकता है. In addition to M13 MP series vector, some other vector like PEMBL or blue script vectors that you may have already studied, they can also be used for this purpose. Dideoxy procedure or chain termination procedure, it is a procedure where during the elongation step of the DNA in the sequencing procedure, the elongation or the addition of further bases, it is stopped. The reason is that in case of dideoxy method, we are although we will use deoxynucleotide triphosphate that are normally present in the DNA, but in addition to that, dideoxynucleoside triphosphate, they are also used. And here, during the sequencing process, we rely on the two capabilities of DNA polymerase. First capability is that it can synthesize a faithful copy of the single-stranded DNA template, so that second strand of the DNA is produced. And the second ability is that in addition to normal base, it can also introduce dideoxynucleoside triphosphate. So this modified base, it can be used as a substrate and inserted during the elongation process. But as soon as it is inserted, the further addition of nucleotide, it is stopped. The reason is that at the 3' of dideoxynucleoside triphosphate, the OH group is missing. That will not catalyze the next reaction. So because of this reason, in the newly synthesized DNA fragment, the addition of new nucleotide, it is stopped. In chain termination procedure, mainly we use Klenow fragment of DNA polymerase. And this Klenow fragment, it lacks 5' to 3' exonuclease activity. And of course, for the initiation of DNA synthesis, a primer is also required. And here primer may be some synthetic oligonucleotide of the DNA. Here it is the comparison of normal deoxynucleoside triphosphate, that is 2' deoxynucleotide. Here OH group, it is not present at the 2' end or site, whereas at 3' location OH group is present in the normal base. When we compare it with dideoxynucleoside triphosphate, then it lacks OH group at the 3' end. So this 3' OH group is missing. That will not catalyze the addition of next nucleotide in the growing nucleotide chain. So in this way, we can use different dideoxynucleoside triphosphates either for adenine, guanine, cytosine, thymine. So we can add modified nucleotide bases so that the chain termination is there after different time intervals. So in case of dideoxy procedure or chain termination method, the DNA synthesis is carried out in 4 different reaction mixtures that contain respective modified nucleotide base, like it may be dideoxy nucleoside triphosphate for guanine or adenine, thymine or cytosine. So 4 different reaction mixture they are used. और जैसा कि मैंने पहले बताया कि हर रिएक्शन मिक्सचर में जब मॉडिफाइड बेस आएगी तो हर स्टेप पे जो चेन की जो एलोंगेशन है वो रुक जाएगी. तो इस तरह से हर रिएक्शन मिक्सचर में हमारे पास डीएनए के फ्रेगमेंट्स आएंगे जो कि वेरी करेंगे लेंथ में. हर किसी में न्यूक्लियोटाइड का जो नंबर ऑफ बेसिस हैं वो डिफरेंट होंगी. व्हाट मे बी द रीजन? द रीजन इज चेन टर्मिनेशन बिकॉज ऑफ द प्रेजेंस ऑफ some modified bases in the reaction mixture. So reaction mixture contain normal bases that belongs to R, all 4 DNTPs and some concentration of dideoxynucleoside triphosphate in a particular reaction mixture. So all 4 reaction mixture, they are carried out or they are processed in the presence of respective dideoxynucleotide triphosphate. When reaction mixture we manage or obtain after the complete reaction, then we will get a mixture fragments that will be separated by using agarose or polyacrylamide gel electrophoresis, and after separation they are detected by a technique which is called as autoradiography. So this is the basic procedure of chain termination method. Suppose here this is the DNA fragment that we want to sequence, and here we have a primer that is complementary with this 3', and so here this is the primer that we will add, and this is the target sequence that we will elucidate in order to get the nucleotide base order. So when the primer is added, then the 4 different reaction mixture they are carried out, either for adenine, cytosine, guanine or thymine. So all 4 DNTPs and their respective dideoxy nucleotides they are added. So this amplification is performed in the presence of cleno fragments, and here we have different fragments of the target sequence, like here 2 guanine are there. So here we have 2 guanine residue, so we will get 2 cytosine at the end, 2 fragments of the cytosine, and in this way different fragments for different bases, like 2 for guanine, 3 for cytosine and 2 for thymine, they are being obtained. So in this way we will get a mixture of different fragments that will be resolved on gel electrophoresis. So in this way we will get a complete sequence of the bases. And finally, this is the polyacrylamide gel autoradiogram that can show the sequence of different base pairs.